Right, so Marcus, again from the University of Liverpool, very complimentary work in partnership with Maya, talking to us now about AI more on the lab side. Marcus, you've got 25 minutes, and I'll jump up and down quite a bit.
Great. Thanks so much, Julian. Okay, so my talk follows pretty much straight on from Maya's work. So what Maya's talked about is what predictions we've got so far and what we've done at the moment and how we're able to predict the potential host range of the viruses that we put in. But I want to talk about is how can we do it better? What can we give the prediction algorithms in order to improve their ability to predict? And that's mostly through either acquiring better data or more data, or generating those data where we can. So a quick recap. I'll give you a quick overview here. So the first thing I was going to do is give you a very quick recap on what Maya talked about. As I follow it straight away, I'll just gloss through that. Then talk about the sort of the general importance of data. I say data are gold, but not all data are equal. Some are a lot more useful than others in prediction problems. So I'll talk about how we can find out which types of data those are and how we can get them. So the two I'm going to focus on are negative data, which is something that nobody really works with very much at the moment, and we want to start working with it a lot more. And then also a technique called active learning, and adding these data that we can get from active learning as well. Right, so as Maya mentioned, we host the EID2 database in Liverpool, and that is essentially where we got our positive data from. So it is a database of interactions between viruses and their hosts. It does more than that, but that's what we're using it for here. And then what we've used that, so if you look at A here, so this is kind of the state of play, as Maya said, what we've observed in the field so far for coronaviruses and their hosts. And then D is what we predict might be able to be the case in terms of viral sharing between hosts and their viruses. So one of the important things to note that from the work that Maya was talking about was that what we'd used so far is just positive data. So just like data from the EID2, where we've looked at which viruses can get into which hosts. And then everything else is considered an unknown. It might be negative, it might be positive, we don't know, it's unknown. We didn't use any information on negative associations. I'll get into a bit more detail of what exactly I mean by negatives in a moment. So essentially what we've done, as Maya said, is we've used the 0.2% of observed positive interactions to predict what's going on in the remaining 99.8. Now, a lot of those are obviously negative, but we've predicted all of them, either positive or negative. So what we want to do is do that better, particularly for the negative side. We want to know what hosts can't get what viruses. And as I say, the two means that we're going to do that by are I'm going to talk through here. Okay, so I think I've just mentioned, or Maya mentioned, that all machine learning based predictions, they require a lot of data. It has to be clean, it has to be up to date, and more always better. But as I said, not all of these data are equal. So one of the particularly useful types of data, especially when you're thinking about virus-host interaction, is negative data. And one of the main reasons it's quite useful is because there's not a lot of it. So if we can get more of that class of data, then that adds a lot to the prediction. But also it tells us things. If a host cannot get a virus, that tells us something about a virus that you don't get from positive only information. It tells you the limits of the range. What's important is there's no existing repositories of negative data at all. It's something we're working on. We've got some applications in to try and build a database so that we can do this, and I'll talk about that. But just to give you a very, very oversimplistic overview, to sort of get your head around why negative data are useful and comparing them to getting more positive data. So if we consider a very oversimplified prediction system, Maya was talking about sort of 400 or 500 different sets of features, but let's for now just consider one. Let's just consider divergence here in a phylogeny. We want to know, can virus X infect this host? And we currently know its host range are these three guys over here. So can we do that with positive only data? Well, not really. I mean, whatever way you cut that, it's either, it could be yes, it could be no. We don't really know enough. It would be the simple way to put it. So if you do the traditional approach, which is to just sort of go out and look for more hosts of that particular virus, you might get something like this, where you just found two more hosts that can be infected with that virus. Does that help? I mean, I personally don't think that that actually, it does improve your prediction a little bit according to the metrics, but it doesn't tell you a lot more. It tells you, okay, that virus's host range is slightly larger than we thought it was before, but we still don't know where its limits are. So if we compare that to negative data, so we start again from the same starting point, instead of going out and looking for more positive sets of data, if we look through the literature or do some experiments to try and find some negative interactions, we might get something like this. This is obviously a best case scenario, where we've again just added 2 more data points. But what we found there is essentially the limits of infection, or got an idea of the limits of infection of this virus. So now I think we've got a much better idea of, can this virus infect this host? And I think we'll probably say with a reasonable guess that it probably can't, because it's outside of that virus's host range. So what we've done there is essentially added the limits of the host range. So that is, there we go. Intuitively, you can see just from that negative data are much more powerful. If you add that into the whole pipeline that we're using, where we're using lots of different features, so we don't just have this graph for phylogeny, we'll have it for every different set of features. So for all of the biases, et cetera, we will find what the limits are. And so the machine learning will be able to put that together in order to get a lot of different limits on the range of the virus. Okay, so what I'm going to talk about now is actually what are negative data. This confused a lot of people, not least grant reviewers. So I'll talk about what exactly I mean here. So positive data are, well, that's pretty obvious stuff, isn't it? PCR sequencing, serology. Can we find that virus in that host either now or evidence that it was there at one point? And that's all the sort of stuff that we have in EID2. That's relatively straightforward. The second type of data is essentially unknown. So things like statements like we've looked but never found an infection, or to a host is in an area that has that particular virus but it's never got it. This sort of falls in your, well, you can't predict a negative or you can't prove a negative kind of definition. This isn't negative data. This is still essentially we don't know. There's not enough information there to make a call in either way when you have those. So what do I mean by true negative data? So this is the kind of stuff that we want to use. So this is actual positive evidence that a virus X cannot infect host Y. So I've got a few examples of this. So one would be this particular goat encephalitis virus. It cannot infect humans because humans don't have the receptor, and that receptor is physically required in order for that virus to infect the host. So that is a true negative. We know that that virus cannot infect humans or anything else that doesn't have that receptor. So we will call that a true negative. The second one is kind of a little bit wishy-washy as to how exactly that differs from unknown. But let's say a common knowledge or a very substantial research effort. So for example, FIV is quite prevalent in cats. There are hundreds of millions, billions of cats around the world. Many of them cohabit with humans and dogs, and many of them take chunks out of humans and dogs. Yet we haven't managed to get FIV in humans or dogs. So that sheer wealth of research effort would lead us to believe that FIV cannot infect those hosts. So we would also call that a true negative. And then finally, competence or susceptibility experiments, where we, in the lab, infect an organism, infect an insect usually, or an animal with a virus and see if it can or cannot get infected with that. So a bit of pilot data from our rummaging through the literature. We went through, I say we, royal we, it certainly wasn't me, went through just over 2,000 publications to look for examples of this true negative data. And we found within our pilot data, just under 10% of these publications actually contained information on true negatives. So it's not really the case that negatives are never published. Often what this might consist of is something along the lines of, we know the receptor is required, we know this animal doesn't have the receptor. That kind of stuff does appear quite a lot in publications. And so that's kind of one of the sources of these true negative data here. So we estimated roughly that there's something along the lines of 1,200 known true negative associations in the literature that we want to extract. So this it sounds like a lot until you think about it, and then it suddenly sounds like not a lot, because there's thousands of species of animals, thousands of species of virus. You multiply that up, and that's actually a tiny number. But what that also refers to are things like this virus cannot infect birds, it can only infect animals, or sorry, mammals. So that's a lot more than just one negative association. That is a negative interaction between every single one of the birds with that virus. And then similarly, we say something is primate restricted, everything outside of primates will also have its own individual virus host negative interaction. Perhaps the best place to look for this, and where we want to do some initial work, some pilot work, is on mosquito competence. So mosquitoes are very well studied for virus transmission, and what's really nice about mosquito work is refractory or non-competent transmission is very publishable still. So if you look at this heat map, we've got different species of virus and different species of mosquito across the side. All of the orange ones are refractory. This is solid evidence that this mosquito cannot transmit this virus. And then all the blue ones are competent. And we show the same thing on the other side here as a network, just showing the interaction between those, but with a similar color scheme. So of over 5,000 individual competence experiments that have been done, a good 30 or so % are refractory. So this is a great starting point in order to have these positive and negative data that we've got. So we can do that for mosquito work. I say relatively straightforward. There's a relatively limited number of papers that we can work on, but we want to do it on a really, really large scale. So how are we going to extract that en masse? Well, as we said, many papers, they do discuss that the inability to infect or a host range is limited by, say, receptors, for example. And we can extract that using natural language processing. So we can use pre-trained language models, things like SciBERT and TaxoNerd, which can just basically just extract these negative arguments from the literature and then, in an automated way, produce all of these data, which will then, of course, will have to be manually checked. But the bulk of the work will be done automatically. So that leaves us to how we're actually going to use these negative data. So if you think back to Maya's talk, where we're using a positive versus unknown approach, well, we can do 3 different types of interaction when we've got the negative data. We can do the same positive versus unknown again. We can also do a negative versus unknown, so we just dump all of the positive data and look at what limits host range to try and get the negative associations more strongly. Or then we could just dump all the unknowns and we could just do a positive versus negative approach. So what we'll essentially have then is 3 sets of predictions for every individual virus host association, which we can ensemble and just select the best one of those 3, because depending on where the virus host is within the space, we might have more negative data, more positive data around that region. So we will either the positive or the negative one will be stronger and able to produce better predictions. So what we'll get there is a much more solid set of predictions, looking at it from multiple perspectives there. So I'm going to put negative data on the back burner now. I'll come back to it at the end and show how it all integrates together in a moment. But that's kind of what we want to do with negatives and how we want to use them. So the next step is something called active learning. So there's another way of perhaps generating data rather than acquiring data. So we've shown that we can extract data from literature. EID2 is positive, we want to start doing it for negative, but we always want more data, and more data are always better. But one group, especially a small group such as mine, data generation is going to be too slow and expensive for us. And we can do it for mosquitoes, but we can't do it for a lot else en masse. So what we want to be able to do so that we can actually generate enough data to make a dent, because even a large group compared to the entire global knowledge isn't going to produce that much. We want to prioritize the most useful data and work out which sets of interactions between virus and host, if we knew whether or not that reaction was positive or negative, that would give us the most information in areas in which we are uncertain. And then, of course, we're going to need a high throughput method of collecting those prioritized data. So I'll talk to you a bit about active learning now. So it's been used before, but sort of mostly in simulation settings. It's not been used much in wet lab work. It's certainly not been used in a large applied problem like this. So it's kind of, we're bringing something new to the field by using this. So what it is, I'll start with sort of a very, apologies to those in the audience that do machine learning, but a very oversimplification of traditional machine learning. So I can build on that from there. So essentially, what you've got here, so if you imagine this is feature space, it could be any of the features that Maya was talking about, but let's just assume host phylogeny versus virus phylogeny, something simple like that. And we've got all the different possible interactions shown by these circles. So the unknown ones are blank, because we don't know them, and then we've got known positives and known negatives. So if we put that, if we use machine learning to sort of decide what, or predict what is going to be positive and what is going to be negative, effectively it just draws a line. And it says anything on this side of the line it is going to predict as positive, and anything on this side of the line is going to predict as negative. So that's going to be really simple what machine learning does. But then in reality, of course, there's an error bar. There's a region of uncertainty around that, shown by this gray space here. So that's usually sort of around the decision boundary there. And that's where the machine learning is not really sure if it's positive or negative. It's not confident of its call. So it gives you basically low performance metrics on the results there. So once you go from that, what we want to do is identify which of those are the most useful bits of knowledge if we did know them. So which of those white circles, the unknowns, if we knew the true answer, if we knew if they were positive or negative, would that be able to improve all of the rest of the predictions in that space? So let's say the active learning selects those 3 dots and says, I want to know the answer to these. If you give me the answer to these 3 points, I'll be much better able to give you a decision boundary and give you a smaller error around it. So those are your high priority data that you want to collect. So to show what we do with that, this is the same picture again as the selected data. We would then do some sort of wet lab testing that we'll talk about in a second. And then once we've acquired those bits of knowledge, we will be able to put them back on the graph, and we'd have a new data set. So back in the database, we'd have a new data set then, which has those particularly informative pieces of data within them. So the first thing you'll notice, you probably can't see it on that small one, but if we zoom in a little bit, you'll notice that a couple of them were wrong. And that's obviously going to happen. We're talking right against the decision boundary, right in a region of uncertainty. It's not going to get every call right. So some of these are going to be the opposite of what the machine learning actually thought it was going to be. So, but that's not a problem. That's almost kind of what we want. So we would then run the whole set of data now with those new data back through the exact same pipeline. And it would, as you can see on the right there, redraw the decision boundary so that it correctly covers where the positives and negatives are. But I think the most important difference there is that the region of uncertainty is narrowed quite a lot. So we've essentially got less error because we've selected the most useful data to use. So this is the before and after what you started without the selected points. So you can see it theoretically at least results in a much better set of predictions after you've provided it with the active learning data. But there's a bit of a problem with that. I mean, let's think about how we're actually going to do that work. What are we asking it to do? And the question there is, can virus X infect host Y? So that was this wet lab testing bit that I just glossed over. That's actually quite a complicated part. It's relatively straightforward in mosquitoes, but outside of mosquitoes, it's a much harder thing to do. So what, for example, if the machine learning wants to know, can a tiger be infected with influenza A? Well, you can't really go out into the field, sneeze on a tiger, come back a week later, jam a cotton bud up its nose, and then see if it's got influenza A. I mean, you could probably send PhD student, postdoc, nobody cares about postdocs. We can lose a few of those, and that'd be fine. But we'd run out pretty quickly. But it could be worse than that. What if it wants to know if an anteater can be infected by Ebola, for example? We certainly can't go into the field and start throwing Ebola around, because that's going to kill more than postdocs. So people are going to get upset. So the issue there, or many issues there, we have with this type of work, so ethics, cost, time, danger. It's basically a no-go. That is lab work that we can't do. We can do it with mosquito a virus infection. It's very straightforward and that's a great model, but we want to do it for more than just mosquito work. So can we do something instead? So it needs to be ethical, of course, not kill anybody, not introduce viruses into the wild. It needs to be low income, or sorry, low cost, because we don't need to do a lot of them, high throughput. We need the results quickly, and it has to be safe. We are working with viruses at the end of the day. So I suppose the ultimate question then is, what determines whether a virus can infect a host or not? This is for a somewhat oversimplified view of this, but for the vast majority of viruses, it's basically a single receptor, a set of receptors, a few receptors, and then a receptor binding protein, and whether or not they attach and fit, that is kind of your main determinant of whether or not a virus can infect a host. Obviously, it gets more complicated than that, but we'll come to that in a moment. But for this first one, this kind of main step, can we make a proxy of it? Well, yeah, of course. There's pseudotype virus particles. We can test this in a lab. We could produce these non-infectious viruses where we can, I'll click on to the next bit, where we can basically change the RBD on the virus by just swapping out the envelope vector, and then so we can create essentially a panel of different viruses or different virus receptors, which we can then test against cell lines, either natural cell lines, modified cell lines, or we're putting different receptors on them. So we can test that interaction relatively straightforward in the lab and do that part of it. And then this is, of course, cheap, ethical, safe. It's a high throughput. It also gives you a much higher sample size than just going out into the field and sneezing on tigers. So it's a much better system of being able to collect the data that we need. But of course, virus host range, it's not all about receptor binding. There are other factors too. So consider things like, does the virus have the ability to replicate inside that particular host species cell? Does it cause too much of a host immune response, for example, and things like that. But these can also all be tested in the lab as well. And we could use the exact same pipeline in order to predict these phenotypes. So we're not just restricted to host range. We can also run the whole thing, but from the perspective of can that virus replicate inside the host species cell too? Depending on your wet lab method, or if you just want to use the more straightforward PVP system, then you can do that relatively cheap and high enough numbers in order to get the data that you need. So in summary, kind of a conclusion to show how all of this works together in order to improve our predictions as much as possible. So that's a set of predictions. I suppose that's not really the first step, but I did put it first on the slide. So the first step is we have the positive data from EID2. That goes in and we can create some predictions from that. We can do some work from that. Maya and I have done quite a few studies on coronaviruses, pox viruses, and then mammalian viruses with that alone. But in order to improve that, we can add some negative data, which is a database that we want to get off the ground to fill in the known data on the other side, the negatives in red there. And then finally, if we can add in this active learning where we would, once we've built as much as we can from the pre-existing data, we then want to produce our own data, but prioritize that data generation by using active learning, selecting those 3 yellow points there, do the wet lab testing, and then go back, put those data back in, and that would then create this cycle where we can do this iteratively, multiple times, each time selecting the most appropriate new data or most informative new data to improve the model with each time. And then, of course, more positive and negative data is coming in fairly continuously. It's not a feedback. So that's the method that we want to use in order to be able to predict host range much better than we currently can. And then to kind of answer one of Julian's questions that he asked a little bit earlier, it also gives us a means to test our prediction. So of course, we're not just going to be selecting things that are right next to the decision boundary. We're going to do some controls on the positive side and the negative side as well. So things that we've said that we're pretty certain are positive, well, let's test those things that we think are positive, just to make sure that, firstly, our wet lab testing system's working, but also the machine learning pipeline is not just spewing out nonsense that we're just making a picture with and assuming is correct. So we need that testing feedback. Okay, so finally, I'll finish up with a thanks. So I've got some collaborators with Maya Dallin, also Andrea Morano, who people who work with us on the computational side from Italy, and Bill and Georgios, some guys from Liverpool who do some of the wet lab PVP work, and then Matthew and Andy. There's no epidemiologists in my group, but the rest of the people from computational and wet lab all work with either myself or Maya in our groups. And then those are the funders who so far have been pretty generous with funding our work. It would be lovely if they were a bit more generous and continue to give us some money. But that's the name of the game, isn't it? Okay, so thanks very much. And any questions?
Thank you very much.
Thanks, Julian.
So if open up the questions, both of them.
Lovely talk. I don't understand the wet lab testing. I'm too close to the front.
Is it you? Is it not me?
Yeah. The wet lab testing, is the plan to apply that to a fairly focused group of viruses where you think you know what the receptor is?
Yes. So if you could imagine sort of every different possible virus and host species interaction, you have this enormous number. Obviously, some of those we're not going to be able to test because, say, for example, we don't know what the receptor is, or we can't get access to something, or say the host species hasn't been sequenced. But because the sheer number of unknowns, and for active learning to work, you need sort of less than 1% of reasonably high priority interactions. We don't have to select all of the top ones. So, if say that the top 1,000 that come up, we can only select, say, 20 or 30 within that top 1,000. That's still more than enough to get the active learning working. So it's basically we're organizing them as a list priority, and we're doing the top ones that we can. But that's sorry to add a little bit more to that. That's for kind of PVP testing. But for mosquito testing, sort of doing it in that system, we can do it for any. So you can just get any old mosquito virus and any old mosquito and feed the mosquito the virus and then do the competence testing that way. So this is why it's a nice sort of starting point, is that we can use these weird combinations without actually having to know all of the sequences, receptors and everything, because we can just test it and the weird thing in the lab.
It's a sort of matrix of pseudotype virus against cells engineered to have the receptor.
Yeah, to do it that way. Yeah.
I would jump in there and say, if you think of some of the jumps that occur, it's because the virus has gained receptor usage.
Yeah.
So you would look at, say, feline parvovirus and go, that won't infect dogs. But feline parvovirus was able to infect dogs because its co-protein changed to amino acids, and then it had a new host. So is there a way you can look at both receptor competence and you've got to distinguish between transmission and infectability of the cell?
Yeah.
So is there a way of also saying, if this virus was to get in this cell, it would replicate?
Yes. So if you well, kind of. To start with the first part, if you say got multiple different genotypes of virus, for example, you could am I too close to the?
No, I'm listening.
Sorry. If you have multiple genotypes of virus, you could basically treat them as different species, different inputs into this. So you could compare one closely related strain to another closely related strain, and they could potentially give different answers. And then you could use, as Maya was talking about, the SHAP values in order to determine which features of that virus were important in that prediction. But then secondly, the wet lab work, you can break that down into the separate components. So you wouldn't actually need to,
All right,
Okay, very quick question. Nice talk. Just one comment before you start infecting postdocs with Ebola. I think next week is postdoc appreciation week. But beside that, one thing you might want to think about, and I don't know if you have thought of this, is looking at your negative data. A source for that outside of publications could be PhD theses.
Yes,
because they're publicly available. They haven't been peer reviewed if they're not in the literature, but they've been peer reviewed by 2 examiners. And you've usually got a huge number of cell lines that somebody's made pseudotype X or Y, and they've tested them against sometimes 30, 40 cell lines. And all of those things may not make it into the publications, but they could feed your system quite well, I would think.
That's a great idea, actually. So the main requirement for us would be publicly available and be extracted from the internet.
I
mean, I suppose, Maya, we because currently for particularly closed access papers, we're just using the title and abstract, which
Okay.
isn't wonderful. Well, most papers now are open access, so we've got
Yeah.
the full data. But we could do it for the whole thesis, couldn't we? I hadn't actually thought of including that, but I don't know if you had.
Because if it's not on an embargo, if it's,
Yeah.
you know, you make them available straight away, they're available from the university repository. And
That's a wonderful
Yeah.
idea. I like that. That's great. Thank you.
Yeah.
Yeah.
welcome.
Thank you very much, Marcus. Thank you.